Hello, I'm Ahmed Abdullah, one of the co-designer of this software tool, BioCluster, which is a quick tool for clustering and identification of antrovector based on biochemical properties. Now, I'm going to introduce you about how to use this tool using some very simple examples. We can run the program by double-clicking the biocluster.exe, the executive file, but it will require MATLAB compiler 7.17 already installed in the system. If we don't have the compiler already installed, we can download it from MathWork website, or we can simply Google it. MATLAB compiler 7.17. This is here is the compiler 7.17. You can either download 32 bits or 64 bits, depending on your system. We'll download 32 bits. Actually, we have already downloaded the compiler. It's here. MCED R2012A Win32 installer. We'll just install this. Okay, so just click next. We'll just follow the instruction. The installation of MCED is complete. MATLAB compiler. Now we can run our program. Okay, this is the page you'll be led to when you double click the executive file. It has 3 tasks available, and they can be navigated through the steps. First, I'll show you how to prepare the data. Then we'll explore these different tasks. I've already some data copy and paste it in the Excel sheet. These are the rows of the data represent the isolates, and the columns represent different biochemical tests. For here, isolate 1 is negative for indole. Negative is denoted by 0 and positive is denoted by 1. To remember that. And all data should be formatted in this manner. 0 denotes negative and 1 denotes positive. That's the convention of this. That's the convention adopted for the software. So these rows are isolates and columns are different tests, and there are 47 possible tests. So first we have to copy this data. And what is this NaN here? NaN is stands for results not available. So we may have an isolate that somehow we don't have a result for a particular test, and it will be represented by NaN. We'll copy this data and let's start with traditional hierarchy clustering. That's the simplest of all the three tasks. Paste that in the input biochemical data edit box, and that's enough. Other options um, are not mandatory. Hit the make dendrogram button, so we have a nice dendrogram. We see different colors in this dendrogram. Oh, first of all, we haven't given the input names of the isolates, so they'll be represented by the isolate's name will be represented by the row number. That's the default option. We can give copy this isolate name and paste it here, and I can hit again make dendrogram button. Now we see that the numbering is replaced by the labels of the isolates. In this dendrogram, this is the distance. For traditional hierarchical clustering, this is actually the Hamming distance, the percent of position different between the isolates. That's the Hamming distance in the x direction. And the coloring and this clustering cutoff is responsible for this coloring scheme. It is for visually identifying the cluster. It makes visual identification of clustering a lot easier. Let's experiment with it a little bit. So, we have clustering cutoff of 0.3. That means it's around here. Anything below this value will be considered a single cluster. As we can see, we should not observe any color above 0.3, and there is none. You see, there is none. Okay, so this is how the clusters are identified. Okay, so let's set it a bit lower. Let's set it here. If we set it here, then this color will also disappear because we're setting it 0.16 perhaps. Then this color will disappear. Let's have a look, and this will also break down to two pieces. Okay, I'll check it. Okay, I see it's lowered. The coloring is lowered. Yes, this thing was a whole single color, and it is split to two different color now. We can give very low value like 0.01, then the all isolate will be different because that's sufficiently low. Not all isolate, this isolate has zero Hamming distance. There will be still a cluster, but let's have a look. There's numerous clusters now. Single isolates are considered a cluster. And if you give a very high value, like here, like 0. anything bigger than I guess 0.45, then it will be the whole dendrogram will be considered a single cluster. Here we see it's a single color. The complete dendrogram, a single cluster. Okay, that's all there is to hierarchical clustering. We can move to the next tab. This warning message asks me whether I want to save my data or not. If you want to save the data, we can do that by clicking show in new window, going to file, to save. You can save as .fig file that will preserve this data and also a lot of things that we can handle with the MATLAB software. .fig is the MATLAB format actually. So we can open it using image viewer. There's a different format also, like .pmp and more conventional format .png, JPEG also. Okay, we can save it if we like. So let's move to the next tab. Yes. Okay. It's similar, but we have to specify additional things in improved hierarchical clustering. We have to copy this data. You have to notice we're not copying the isolate names or the name of the test. We're just copying the data and input by pasting it here, and then let's copy this isolate names. Then we have to select the test, select the name of the test from the list, select test box. We have I guess indole production, methylred, Boscowers, citrate, hydrogen sulfide. I guess I'm correct. No. Yeah, citrate, yeah. Then I have dextrose, lactose, and sucrose. Dextrose is D-glucose. It's acid formation test actually. In this case, I also need lactose and sucrose. Okay. We have to give additional information to this to the program for improved hierarchy clustering. That is, you have to inform the program the sequence of the test as they appear in the biochemical data. We have selected the test. The program knows that this is the test, but in which order they occur in the biochemical data edit box. What this column represent in terms of the test? Is it indole? Is it methylred? What is it? What the second column represent? What the third column represent? One neat way to arrange the data is to follow the sequence in which the select test list box is arranged. Namely, we can put the first column as the indole production, the second column as the methylred, and so on accordingly to this list box. If we arrange this data in that manner, we don't have to actually provide any sequence. The program will take that as a default sequence, as the sequence appear in the select text box. Okay, then actually these data are arranged in that manner, so we can simply press the make dendrogram button to generate the dendrogram. We have to give additional information to this to the program for improved hierarchy clustering. That is, you have to inform the program the sequence of the test as they appear in the biochemical data. We have selected the test. The program knows that this is the test, but in which order they occur in the biochemical data edit box. What this column represent in terms of the test? Is it indole? Is it methylred? What is it? What the second column represent? What the third column represent? One neat way to arrange the data is to follow the sequence in which the select test list box is arranged. Namely, we can put the first column as the indole production, the second column as the methylred, and so on accordingly to this list box. If we arrange this data in that manner, we don't have to actually provide any sequence. The program will take that as a default sequence, as the sequence appear in the select text box. Okay, then actually these data are arranged in that manner, so we can simply press the make dendrogram button to generate the dendrogram. There is additional options called the distance metric. We can choose either Bayesian probability distance, or we can choose Bayesian cost function. Okay. Let me first explain if the data are not arranged in as they appear in the select test list box, how should we provide the sequence information? We should ask this question. For the first position, we'll say that where indole production appear in the data, in which column indole production appear in the data. Let's see here. It's actually in the first position. Let me just change the order for the purpose of illustration. Okay, I've changed the order. Now indole appear in the 8th column, and meteorite in the 2nd, Bospascaus in the 3rd. Where does indole production appear in this data? Okay, I have to copy this data actually. Okay, this is my newly arranged data. Where indole production appears in this data, it appears in the 8th column. Okay, so we'll write 8 here. Where methyl red appears in this data, let me check. Methyl red appears in the 1st column, so I write 1 here. While Voges-Boscauers appears in the data, it appears in the 2nd column. Where citrate appears in this data, citrate test, it's appears in the 3rd column. Hydrogen sulfate in the 4th column, we already know that. 5th, 6th, 7th, because we have only messed around with the indole production. The rest remains the same. Okay, then we can hit the dendrogram button. Oh, we can choose the distance metric. Let's choose the Bayesian probability distance. We hit the make dendrogram button. Okay, now we have the dendrogram generated. We can choose the distance metric, Bayesian probability distance, or Bayesian cost function. Which distance metric should you choose? Bayesian probability distance is good for clustering. The same kind of organism will be clustered together. Not same kind of, actually, same organism from the same species will tend to cluster together in the Bayesian probability distance. It's best at doing that, and Bayesian cost function is capable of reflecting the phylogenetic relationship between the species. It's not best at grouping, but it's best at capturing the intergroup relationship between species relationship. So I think I should select it. We can choose probability distance first in order to identify the clusters. Then later on, I think we can choose the Bayesian cost function to choose the relationship between the groups, but we have to take the support the supportive information from the Bayesian probability distance in order to get the clusters right. And we have also other button called showing entry vectoricity. This what this button. This button shows the data uh, in shows the position um, of our isolates with respect to Enterobacteria randomly generated Enterobacteriaceae species. So these are randomly generated Enterobacteriaceae species, and these are our isolates in the middle of some species. It will help us to visualize our position of the isolates in this tree. You can choose based on cost function also. The tree will be generated using this distance metric or that distance metric based on cost function. It takes a little bit more time because it's the algorithm is computationally expensive. We see isolate 2038 here with Serratia odifera biogroup. Okay, let's move to the next tab, which is called the identification tab. Oh, before moving, we can use this tool to aid our visualization. We can, for example, drag the data if we like. We can zoom and zoom out. I think at this moment I need to zoom in. Yes, I have zoomed in. I can also look at the different data point. X, Y. X value is irrelevant. What's relevant is the Y value actually. And we can rotate the dendrogram. We can zoom out and rotate. Perhaps it is helpful for a smaller tree. It will also be helpful for recognizing the topology of the tree. Okay, let's move to the next tab called the identification tab. Okay, done. Identification tab has a similar pattern as the improved hierarchy clustering. We have to provide the biochemical data, we have to give the name of the isolates, we have to select the test, and we have to provide the sequence of the test as they appear in the biochemical data. And select the test. All are the same test. Lactose, sucrose, and we have to select the test sequence of the test. And we can press the button identify. So identify button will produce the Bayesian probability of species will produce the probability of a particular isolate to belong to certain Enterobacteriaceae species. Okay, and we can choose the number of result visible under a isolate. We can provide this show the best match of the first. We can provide it any number. We can provide 10. Then 10 result will be shown. The best 10 result will be shown. That isolate 1 has the probability to belong to Providentia alcani faciens 0.24948. that is the probability that isolated belongs to this group, and so on. And we can choose any, we can choose 5, for example. Okay, and we can also uh, visualize the position of our data in the anti-recursive tree. There is no distance metric. Actually, cost function is chosen as the appropriate distance metric here. Okay, we can see our isolates, isolate 1, 2, 5, and 4. I hope this program will help you to understand the operation of BioCluster tool. Thank you for watching. For more information, visit our website at www.microbialgen.tu.ac.pd.